The evolution of color vision in primates is unique compared to most eutherian mammals. A remote vertebrate ancestor of primates possessed tetrachromacy, but nocturnal, warm-blooded, mammalian ancestors lost 2 of 4 cones in the retina at the time of dinosaurs. Most teleost fish, reptiles and birds are therefore tetrachromatic, while all mammals, with the exception of some primates and marsupials, are strictly dichromates. Primates achieve trichromacy through color photoreceptors, with spectral peaks in the violet, green, and yellow-green wavelengths. Opsin is the primary photopigment in primate eyes, and the sequence of an organism's opsin proteins determines the spectral sensitivity of its cone cells. Not all primates, however, are capable of trichromacy. The catarrhines are routine trichromates, meaning both males and females possess 3 opsins sensitive to short, medium, and long wavelengths. In nearly all species of platyrrhines, males and homozygotic females are dichromates, while heterozygotic females are trichromates, a condition known as allelic or polymorphic trichromacy. Among platyrrhines, the exceptions are Alouatta and Aotus, both of which are uniform trichromates. Mechanism of color vision. Genetically, there are 2 ways for a primate to be a trichromate. All primates share an S-opsin encoded by an autosomal gene on chromosome 7. Catarrhine primates have 2 adjacent opsin genes on the X chromosome which code for L and M opsin pigments. In contrast, platyrrhines have only a single, polymorphic X chromosome M/L opsin gene locus. Therefore, every male platyrrhine is dichromatic because it can only receive either the M or L photopigment on its single X chromosome in addition to its S photopigment. However, the X chromosome gene locus is polymorphic for EM and L alleles, rendering heterozygous platyrrhine females with trichromatic vision, and homozygous females with dichromatic vision. Proximate causation hypotheses. Some evolutionary biologists believed that the L and M photopigments of New World and Old World primates had a common evolutionary origin, molecular. Studies demonstrate that the spectral tuning of the 3 pigments in both suborders is the same. There are 2 popular hypotheses that explain the evolution of the primate vision differences from this common origin. Polymorphism. The first hypothesis is that the 2-gene system of the catarrhine primates evolved from a crossing over mechanism. Unequal crossing over between the chromosomes carrying alleles for L and M variants could have resulted in a separate L and M gene located on the single X chromosome. This hypothesis requires that the evolution of the polymorphic system of the platyrrhine predates the separation of the Old World and New World monkeys. This hypothesis proposes that this crossing over event occurred in a heterozygous catarrhine female sometime after the platyrrhine-catarrhine divergence. Following the crossing over, any male and female progeny receiving at least one X chromosome with both M and L genes would be trichromates. Single M or L gene X chromosomes would subsequently be lost from the catarrhine gene pool, assuring routine trichromacy. Gene duplication. The alternate hypothesis is that opsin polymorphism arose in platyrrhines after they diverged from catarrhines. By this hypothesis, a single X opsin allele was duplicated in catarrhines, and catarrhine M and L opsins diverged later by mutations affecting one gene duplicate but not the other. Platyrrhine M and L opsins would have evolved by a parallel process, acting on the single opsin gene present to create multiple alleles. Geneticists use the molecular clocks technique to determine an evolutionary sequence of events. It deduces elapsed time from a number of minor differences in DNA sequences. Nucleotide sequencing of opsin genes suggests that the genetic divergence between New World primate opsin alleles is considerably smaller than the divergence between Old World primate genes. Hence, the New World primate color vision alleles are likely to have arisen after Old World gene duplication. It is also proposed that the polymorphism in the opsum gene might have arisen independently through point mutation on one or more occasions, and that the spectral tuning similarities are due to convergent evolution. Despite the homogenization of genes in the New World monkeys, there has been a preservation of trichromacy in the heterozygous females, suggesting that the critical amino acid that define these alleles have been maintained. Ultimate causation hypotheses. Fruit theory. This theory encompasses the idea that this trait became favorable in the increased ability to find ripe fruit against a mature leaf. Background. Research has found that the spectral separation between the L and the M cones is closely proportional to the optimal detection of fruit against foliage. The reflectance spectra of fruits and leaves naturally eaten by the Alouatta seniculus were analyzed and found that the sensitivity in the L and M cone pigments is optimal for detecting fruit among leaves. While the fruit theory holds much data to support its reasoning, recent research has gone on to disprove this theory. Studies have suggested that the cone pigments found in dichromates can actually distinguish the color differences between fruit and the foliage surrounding it. Furthermore, the survival benefits favoring the selection for this trait are not completely necessary, as many trichromatic primates live in environments where fruits have similar color tones in relation to the surrounding foliage, and yet studies have shown that these primates obtain the same proportion of fruit compared to their dichromatic relatives. Young leaf theory. This theory is centered around the idea that the benefit for possessing the different M and L cone pigments are so that during times of fruit shortages, an animal's ability to identify the younger and more reddish leaves, which contain higher amounts of protein, will lead to a higher rate of survival. This theory supports the evidence showing that trichromatic color vision originated in Africa, as figs and palms are scarce in this environment, thus increasing the need for this color vision selection. However, this theory does not explain the selection for trichromacy polymorphisms seen in dichromatic species that are not from Africa. Long-distance foliage hypothesis. This hypothesis suggests that trichromacy has evolved to adapt to distinguishing objects from the background foliage in long-distance viewing. This hypothesis is based upon the fact that there is a larger variety of background S and luminance values under long-distance viewing. Short-distance foliage hypothesis. This hypothesis suggests that trichromacy has evolved to show higher sensitivity to low spatial frequencies. Spatiochromatic properties of the red-green system of color vision may be optimized for detecting any red objects against a background of leaves at relatively small viewing distances equal to that of a typical grasping distance. Evolution of olfactory systems. The sense of smell may have been a contributing factor in selection of color vision. Studies suggest that the loss of olfactory receptor genes may coincide with the evolved trait of full trichromatic vision. In other words, as sense of smell deteriorated, and thus the ability to identify the most vital nutritional sources, the necessity for advancement in other senses increased, and the likelihood for trichromatic color vision mutations to remain selected for became higher. In addition, the mutation of trichromacy could have made the need for pheromone communication redundant and thus prompted the loss of this function. Overall, research has shown that the concentration of olfactory receptors is directly related to color vision acquisition. Interestingly, research suggests that the species Alouatta does not share the same characteristics of pheromone transduction pathway pseudogenes that humans and Old World monkeys possess, and leading howler monkeys to maintain both pheromone communication systems and full trichromatic vision. Therefore, trichromacy alone does not lead to the loss of pheromone communication, but rather a combination of environmental factors. Nonetheless, research shows a significant negative correlation between the two traits in the majority of trichromatic species. Health of offspring. Trichromacy may also be evolutionarily favorable in offspring health through mate choice. M and L cone pigments maximize sensitivities for discriminating blood oxygen saturation through skin reflectance. Therefore, the formation of trichromatic color vision in certain primate species may have been beneficial in modulating health of others, thus increasing the likelihood for trichromatic color vision to dominate a species' phenotypes as the fitness of offspring increases with parental health. Anomalies in New World monkeys. Aotus and Alouatta. There are two noteworthy genera within the New World monkeys that exhibit how different environments with different selective pressures can affect the type of vision in a population. For example, the night monkeys have lost their S photopigments and polymorphic M.L. opsin gene, because these anthropoids are and were nocturnal, operating most often in a world where color is less important. Selection pressure on color vision relaxed. On the opposite side of the spectrum, diurnal howler monkeys have reinvented routine trichromacy through a relatively recent gene duplication of the M.L. gene. This duplication has allowed trichromacy for both sexes. Its X chromosome gained two loci to house both the green allele and the red allele. The recurrence and spread of routine trichromacy in howler monkeys suggests that it provides them with an evolutionary advantage. Howler monkeys are perhaps the most folivorous of the New World monkeys. Fruits make up a relatively small portion of their diet, and the type of leaves they consume are best detected by a red-green signal. Fieldwork exploring the dietary preferences of howler monkeys suggests that routine trichromacy was environmentally selected for as a benefit to folivore foraging.